I would like to ask you if you can take your seat, and then we would like to be on time. So I would like to start this session on the research for biotech innovation. First of all, I would like to thank to the organizer for organizing this event and put us all together, and also for giving me the chance to be a chair of this exciting session on the research for biotech innovation. So I would like just to present a few slides before our speaker. So I would like to ask you if you can put our presentation. Yes, so let's see. So we will all agree that every aspect of our professional, personal life have been touched by COVID-19. So pharma and biotech companies have shown an incredible response to the COVID-19 pandemic. Resources have been strategically allocated to research efforts in order to provide therapeutic solutions in a timely manner. So I just wanted a brief introduction as a chair of this section to say how COVID-19 shaped the research for biotech innovation. So almost all other research has grounded to a halt during the changing priorities and social distancing measures that restrict access to laboratories. So that was the beginning of COVID. So as the result, many scientists from many different fields are now involved in the study of SARS-CoV-2, including myself and our lab. During the COVID-19 pandemic, large number of biotechnology companies shifted their focus to infectious disease research. Online repositories make studies available months before they are published in scientific journals, and we saw that within 60 days researchers have sequenced and shared hundreds of viral genomes and published more than 4,000 new studies relating to SARS-CoV-2. While most countries have closed their borders, scientists open theirs, bring about global collaborations of an unprecedented scale. So many multidisciplinary teams, consortia of research centers, university laboratories, hospitals, vaccine manufacturers in different parts of the world are emerging. Biotech companies started clinical trials on potential treatment of COVID just 3 months after the genetic sequence of the virus has been made published. So if you look at this graph, you can see that it was much faster than when 2003 the first SARS outcome came out. So this is the duration of on this graph you can see the duration of biotech companies respond to COVID-19 pandemic.
Quindi molto più veloce rispetto al 2003, quando ci sono state altre minacce sanitarie e altri focolai fra il 2003 e il 2009. Alle risposte del Biotech alla pandemia del Covid-19 noi abbiamo visto che si è trattato di collaborazioni in tempo reale fra scienziati in tutto il mondo in una corsa per sviluppare la produzione di vaccini e farmaci antivirali. Fino al 5 gennaio 2021 c'erano 191 candidati come vaccini Covid-19 in corso e oltre 800 studi clinici per vari farmaci e vaccini, molti dei quali sviluppati da aziende che non avevano precedente esperienza nel settore della malattia infettiva. Si trattava veramente di una vera risposta alla pandemia del Covid-19. Il settore industriale ha trasformato e convertito i processi di produzione per produrre forniture di ambito medicale su larga scala, e anche le aziende locali e nazionali hanno reagito in tempi molto brevi, modificando non solo la produzione ma anche i processi di ricerca e sviluppo. Gli innovatori tecnologici e le start-up si sono uniti per fornire soluzioni alle problematiche più urgenti. Ne abbiamo sentito parlare tutta la mattina di queste squadre, di questa collaborazione. E chiaramente l'avere dei forti rapporti di fiducia fra le singole aziende farmaceutiche ha rafforzato la velocità e l'agilità della risposta dell'industria nel fornire farmaci e vaccini, il che ha accelerato non soltanto i trasferimenti di tecnologia con collaborazione a più attori ormai formalizzate. Per alcune aziende la capacità di produzione è più che raddoppiata dall'avvio della pandemia. Pertanto volevo soltanto dare una breve panoramica di questa innovazione nel contesto del Covid-19. Innanzitutto abbiamo sentito parlare di drug repurposing, che è stato particolarmente interessante considerando che la tossicità di alcuni composti era già stata testata. Poi abbiamo acquisito nuove conoscenze nell'ambito del medical imaging, del potere di calcolo e delle operazioni cliniche, il che ha accorciato la durata degli studi clinici, dando una maggiore conoscenza e comprensione dei dati, garantendo un adeguato sviluppo e l'applicazione di prodotti terapeutici. Abbiamo sviluppato nuovi anticorpi e nuove terapie. Diversi approcci allo sviluppo dei vaccini sono stati approntati, abbiamo avuto nuovi strumenti e nuove tecniche che hanno consentito l'agilità e la flessibilità degli ecosistemi in questo settore. Dopo questa breve introduzione volevo soltanto mostrarvi alcune diapositive per quanto riguarda il mio gruppo e la nostra ricerca nell'ambito del Covid-19, la nostra risposta al Covid nel corso dell'ultimo anno. Io sono responsabile della Biotechnology Development Unit, l'ICGB, questo è il gruppo che si occupa dello sviluppo dei farmaci biologici. Nel corso del Covid abbiamo acquisito una lunghissima esperienza che già avevamo in passato utilizzando varie piattaforme come cellule di mammiferi, lieviti e batteri, ma durante il Covid noi ci siamo concentrati soprattutto sul trasferimento di tecnologia alle aziende farmaceutiche in tutto il mondo. Abbiamo sviluppato le formazioni su base video. Pochi mesi dopo l'avvio della pandemia abbiamo sviluppato i nostri moduli di formazione per poter essere in grado di fornire queste tecnologie tramite questa piattaforma online. Nell'epoca della pandemia abbiamo dedicato gran parte del nostro tempo a sviluppare nuove strutture, nuovi laboratori dove possiamo con una scala migliore, in condizioni migliori, produrre e sviluppare farmaci biologici. Questi sono i nuovi laboratori, siete tutti invitati a visitarli. Sono 11 nuovi laboratori con l'ultima attrezzatura state of the art disponibile per i biofarmaci. A parte questo abbiamo creato un gruppo di virologia di cui sentirete parlare successivamente con Alessandro Marcello, sentirete da lui come gran parte del suo tempo sia stato dedicato alla ricerca sul SARS-CoV-2 nel tentativo di sviluppare dei kit diagnostici per il Covid. Abbiamo reagito con grande velocità, adesso il know-how c'è, invece di produrre interferone e insulina possiamo produrre anticorpi alla proteina spike, farmaci. Questi sono i vantaggi che abbiamo, come la disponibilità immediata, costi ridotti nei nostri laboratori di ricerca all'interno dell'SGB e anche all'esterno, in tutte le entità che Alessandro con cui Alessandro collabora. Anche per loro questi costi sono ridotti. Ecco perché siamo in grado di reagire con grande velocità. Ora, prima di presentare i vari oratori che condivideranno la loro esperienza e le loro reazioni al Covid-19, la loro prospettiva per un post e la loro
Bruno Bembi, who is the CEO of Transactivia, and then we can go to the speakers. I'm speaking in English, but I'm whoever of the speakers that can choose to talk or in Italian or in English. Thank you.
Thank you, Natasha. I just want to say some words about the collaboration we have with the group of the ICGB. That is a collaboration that is aimed to develop a plant molecular farming platform for the production of vaccines. This is an interesting point because it means that we, in the future, can use the plants as bioreactors to produce protein for therapies proposal and particularly for vaccinations. This is something of new. During the COVID era, this aspect broke the, let me say, psychological difficulty to deal with GMO plants. And in the future, I'm convinced that the use of this technology will share by different groups in the world and will also develop in our countries. That is still difficulty to convince authorities and population about the facilities that can be developed with plant molecular farming. I think also that something that we have understand with the COVID era is the psychological difficulty for the population, from part of the population, to accept the science or the scientist as, let me say, team leader in developing political, how to say, choosing in the approaching the healthcare system. So this is something that we have to consider in the next future, and because in general, as a scientist, me personally, I'm sometimes strong against these persons, but I think that we have to discuss and to convince and to improve education and improve the knowledge of science and of scientists. So thank you for the introduction you made, and we have to begin the session.
Thank you very much. So I would like to ask the first speaker, Dr. Alessandro Marcello. He's a group leader at the ICGB. He's heading the molecular virology laboratory, and he will speak about the ICGB, what ICGB did and do in a COVID matter, how the ICGB have responded to the emergency from the biotech point of view. Alessandro, you have 10 minutes, please.
Funziona questo? Sì. Parlo in italiano o in inglese?
Come vuole.
Ah, facciamo italiano, che mi sembra.
Okay, I will use English. Now, the first slide, I always show this first slide because I want to tell my audience who we are. We are an international organization, ICGB, and we are affected by strabismus. We work here in Friuli Venezia Giulia, but then we also address other countries in the world, from Africa to Asia and Central and Latin America. We want to develop biotechnologies that are useful for the whole of our constituents in Italy and outside of Italy. So all our studies and works have these two objects in mind. I am in charge of the molecular virology lab, so I was involved, personally involved in fighting the pandemic, and we started immediately, already in February last year. Is someone, oh, you can do it. You can do it on your own. What did we do starting in March last year? We locally worked with the local health agency and Area Science Park to isolate the virus and our patients to do sequencing of the virus. At international level, we try to develop new biopharmaceuticals. We started repurposing drugs against COVID, but also we wanted to develop new drugs, and then we supported local industry, but not just local companies, to clean up the environment to defeat the virus in the workplace. And then we supported the member states of the ICGB in the fields of diagnostics, sequencing. All our results are available to all our member states. There is a website where we share all of our results. One of the key technology, or one of the key facilities that we have at ICGB is a BLS3 lab that allowed us to isolate the virus, which is fundamental for any kind of advancement, because if we can't have a culture of the virus, we can't do anything at all. But we had these capabilities with the level 3 viruses, so we were ready to work in vivo with the COVID virus. We made an effort in sequencing. Now sequencing now is very much advanced. Sequencing is done very well in Italy, in Europe. Progress was made in the precise sequencing of the genome of the virus. But in March, when we started, in Italy, there were only 3 sequences available. We sequenced the circulating virus, and all the sequences were immediately made available on public databases. This went on over time. We continued to collaborate with local health agency and area. We isolate the virus, and we do its sequencing, which is important because sequencing is important because it gives us an idea of how the virus evolves. But it is also important to distinguish among the different variants, how pathogenic they are, and we have to do that in a lab. By isolating the virus, we can study whether that variant can be inhibited by a certain drug, whether it has become resistant, whether it can be inhibited by an antibody, which is of paramount importance for epidemiological surveillance. Of course, we did not confine ourselves to Friuli Venezia Giulia alone. We implemented other programs with ICGB countries. We helped them sequencing the locally circulating virus, because in Italy and in Europe now we have a good distribution of sequences. We know exactly what happens every day. But in other regions of the world, we don't know really what's going on. I really liked when the variants were called with the geographic area of origin, the Nigerian, the English variant, or the Brazilian one, because the virus is global. And in many countries, there might be different variants. So what counts is not just a European sequencing. We should be doing that all over the world. With Moldova, Cameroon, Ethiopia, Kenya, Nigeria, Santo Domingo, and Iraq, we help them sequencing locally the circulating virus. So we are trying now to help these countries creating local sequencing centers so that they can work with the local situation in a greater vision of, in the greater framework of the epidemiology of infective diseases. As for diagnostics, we worked both at molecular and immunological levels. We worked with the health agency for the ELISA development. With Salus, we assessed various diagnostic kits. With Halifax, we collaborated to develop the molecular mouse for the SARS-CoV-2 and rapid tests, and we continue to work with them. This is what we are doing locally. And internationally, we developed a LAMP test, which is alternative to the classic conventional PCR with the amplification of viral genome. It is based on an easier methodology. We need the method is simpler, and it can be applied in countries where technologies are not that advanced. And in this sense, we collaborate with the Bill and Melinda Gates and with another American company. Development of antiviral drugs. We have high throughput methods to identify new drugs, thanks to the antibodies that we developed in the lab, and thanks to Natasa that purified them. And we've been able to test antiviral drugs. We started with Miglustat together with BioValley. We had that drug available. It was an antiviral for other infected disease, and we found out that it worked against COVID as well. We discovered the activity of the 27-hydroxysterol as an inhibitor together with Panoxivir, which is a spin-off of the University of Turin. We worked with Sun Pharma, which is a Indian pharmaceutical company, to use the AQCH, which is has already the clinical trial phase 3 is already completed. Now we are working on the repurposing of nitroxaline as an inhibitor. This is an example of drugs that worked, and then there are hundreds of them that didn't work, but we performed a screening on random medications, but also on potentially beneficial molecules. And this goes on, not just to identify new drugs, but also to understand how these drugs are working, what their action mechanism is at molecular level, because that helps us in improving them and making more effective. And finally, we collaborated with the industry. This is an aspect of my research that I tended to neglect, but now it has become increasingly relevant, and it became so already at the beginning of the pandemic. The industry asked us to help them to inactivate the virus in the environment. It could be, well, the most important activity was on cruise ship with Fincantieri in large ships to decontaminate the environment, the air, the surfaces on a large ship. So we carried out this test together with Fincantieri. We worked with Luxottica and with many other Italian companies, and not just Italian, but also international ones. And this was a new activity for my lab, but it was very, very interesting and very important as well, because I think that over the next few months and years, we will need these decontamination methods to be massively used in crowded environments. I'd like to conclude because I only have 10 minutes available. I just wanted to remind you that basic research has to be funded. Everything begins with basic research, translational studies that can be further applied to the world of our company. So basic research has to be funded. We did not become virologists March last year. We were virologists before, and we're still virologists. What we need is a know-how that can be applied to many forms of collaborations. Thank you. Thank you, Alessandro Marcello.
Alberi Auber, who is the director of the Swiss Integrative Center for Human Health in Freiburg, Switzerland. And she will talk about clinical diagnostic studies showing the reliability of salivary diagnostics for COVID-19 infections and how this lead to the development of patented method for the molecular diagnostic in saliva. So if Lavinia is ready. So you can change on your, yes, you can sit there and change on your seat.
It's not possible to change
The slides?
I'm not an expert. Maybe you have to send the slides to me.
No, we already have it, no? Easy to...
You have here slides.
I'll try to present that.
Maybe in front of the microphone so they can hear you better. Yes.
Yeah, sorry for that. This has been a very tough day for me because we are still dealing with mass testing exactly with this PCR molecular method that we developed, which is very, very fast. But it's a direct method, and so it does work very, very well with saliva and other biofluid, even cell culture supernatant and other biofluid. But then when you put ions inside, then things work not so well. So with salivary gargling solution, doesn't work well. Unfortunately, this was not completely the presentation I wanted to share with you, because I had many more data, scientific data I wanted to show that proved that our methods is completely overlapping, if not better, than the extraction method. We tested 100 positive patients through a study, which is we're now preparing. And with the values of viral titer, which go with CT values, this is CT, the cycle of the PCR, which are very, very low, about 15 to 37, and we can detect the virus to very low titers. So we are pretty satisfied with that. And then we performed comparison between our method and the presence of the virus of nasopharyngeal swabs with one kit, and they were completely matching. So we have 100% match using the same kit. And so maybe I will start saying that after this clinical validation, we started selling the test. And the test is really a salivary test, so very, very easy to implement. The subject are recognized by a barcode, which is on the test tube. And this barcode follows the patients all through the diagnostic journey. The information about the subject that are also requested by the Ministry of Health are recorded into a sick app. And there is also the number of the insurance, because if the subject is positive, it needs to be reimbursed by the insurance. In Switzerland, the health system is not public, it's private. So everything is reimbursed by the insurance or directly the Confederation, if it's for mass testing. And then the subject book an appointment for the video identification, which is requested by law. Okay, which is requested by law in order to obtain a QR certificate. And then the subject, once he has performed the video identification, which we have developed also in collaboration, that was developed by a SIC app that works with the BioValley group. We implemented it in August, I think, in August. And basically we have the whole journey of the subject remotely. So, for example, if someone orders the test on Wednesday, we will send the test kit on Thursday. On Thursday afternoon, he does the identification with auto collection with the operator, and then he or she just sends the test back to the SIC. The SIC gets the test kit on the Friday morning, and the client gets his results by Friday afternoon. So the whole process is very lean. The tests are of the price of the nasopharyngeal swabs test, so there is absolutely no difference. Of course, if someone is positive, then it will be reimbursed, but the majority of our clients are travelers, so they pay for themselves, if not the two tests that are paid by the insurance company for the holidays. So there is also video here that someone can look at. The method it's very simple. It works basically with this funnel and the tube. I'm looking actually for producer of small tubes which have barcodes. This is really key, because with this barcode you do identify the subject, and the saliva is stable for 3 days at room temperature, as by publication. Again, everything is sterile, and the barcode is the barcode of the subject, and this will be also recorded into the SIC app. So through identification and a barcoding scanner application integrated into the SIC app. So I mentioned that it's completely secure because our video identification runs on a video application that is used already by a general practitioner, and so it's recognized. Of course, you need a smartphone or a PC in order to be able to access the medical assistant and to do the auto collection in front of the medical assistants. In case of mass testing, this is not used. There are medical personnel inside the company, if those are big, and there is an identification made by the personnel inside the company. So the innovation is really in the fact that we can use everything which is already available in the lab. However, we can also use automatization, and we are definitely faster, 3 times faster, and 5 times more efficient, and we are much cheaper. So For example, with the with the extraction method, you have more or less about 9 francs, which is about €7 cost for the extraction. Here you have 0.5, so it's almost 10 times cheaper than the extraction. The clinical validation shows a 98.6% sensibility and 100% specificity, which is inherent to the test. We use a microgen kit, which has 2 genes. We also used other kits with 3 genes, the C gene, and it works as well also with other kit. It's scalable, so we can definitely use automatization. We typically work with pooling samples, especially for the mass testing. We typically, as we are looking for asymptomatic subjects, we do an 8-time pooling that is suitable for automatization. But I think that we can go with actually with breakthroughs. Without breaks, sorry, we can also go into a matrix pooling that we can also organize with the robot.
Lavinia, I would like just to ask you the last minute.
Yeah. So the patented method has been used already. This is actually, it's missing 1 0, but it's 200,000 people we have tested, and we have 20 companies, 15 schools, and 10 pharmacy. And this study was conducted. This testing was made able by the clinical study that we're going to be publishing soon.
Okay. Thank you very much, Lavinia, for this, and congratulations on this amazing work. So I would like to give a floor to Salvatore Guarneri, AD of Sanatorio Triestino in Trieste, so he can share us his experience during this challenging time, being part of a modern hospital complex. Please, this is yours.
Grazie, buongiorno a tutti.
Thank you, and thank you for the invitation. Thank you, organizers, for this very fruitful day filled with interesting observations on a host of topics. We are not on the part of the research, but more of the practical application of some devices and activities. The Sanatorio Triestino institution from 1897. It's quite an old startup, if you will. Some years ago, we saw the archives from 120 years ago. Amongst its founders, there were all university professors, Austrian in origin. Some came from Linz, some from Paris. And one of the articles I found from 1915 was from a Triestine urologist, Nikolic, who wrote an article, also in English, at European in error in urology. So, to write about errors in medicine, with the first publication of American studies where they're signaling that 50% of mortality and errors in a clinic. When I found this article, I was amazed that he was so advanced in time. He was talking about his errors, and he asked everyone to publish, even anonymously, to publish their own mistakes in medicine with technologies that we saw earlier with internet stent law. There is very little space for mistakes. The facility became a company in 1996. It's one of the oldest companies in Friuli and one of those that managed to resist despite the war, and was characterized by surgeries. In the COVID period, all facilities were called upon to offer beds for the COVID crisis, And I like the presentation earlier, which showed the evolution in the last 30 years of the beds in medical facilities and how this has had an impact on the training, documentation, and how this in turn has had an impact on each and every facility from an organizational point of view. And one of the difficulties that we've had is that of evaluating innovation, how to simplify our lives and adapt to innovation. In the COVID period, we had tens of rapid or non-rapid tests in order to identify various types of therapies that were suggested as it has been presented so far. It was one of the greatest difficulties. Selecting the technology is one of our main issues. It's an impossibility of trying them out first. Technology and innovation within a facility has areas and enthusiasts in adopting new technologies and the naysayers against it. And the difficulty is almost on a daily basis because there are so many technologies that it's difficult to evaluate it properly. The devices that we had for identifying COVID, identifying the infection, were stored in the ministry the day before. We got the documentation from China, from any part of the world, but it was impossible to verify its functionality. So we just looked at the documentation and accepted making choices with a blindfold because we were not able to check them. This was one of our greatest issues. Luckily, I was managing the Quiete in Udine and the Sanatorio with 150 patients who are not autonomous. We did not have any infection in 2020, and I don't think it's a matter of luck. Whereas in the Sanatorio, we created a COVID area for positive patients. We managed with the tools at our disposal to keep the infection under control in some areas and guarantee or reduce what could have been its spread. One of the main problems we found while in the Udine facility, we had a software for the medical files, medical records, which allowed us to go less in a certain ward or room and have a dialogue, a distance with our operators. In February 2020 is where we started. To be honest, we actually started scheduling these activities before COVID. I want to talk about medical records because, in my opinion, it's one of the instruments that brings together knowledge and technology, and which is not that well done as the market would require. The integration among software is possible nowadays. Streamlining the operators in order that they have the choice or be driven towards a certain choice. Software nowadays shows only a collection of chaotic data. Nobody is investing to reorganizing the numerous data that all of us have in order to streamline us towards a certain clinical choices. So we have an iTeam system which is outdated, which we had in radiology wards many years ago, but this does not communicate with archives. We have an archive, but no organization on pathology, X-rays. The technology has moved so far ahead because you have more than 5,000 sections for a medical test. maybe the physician could organize some sections as he wants, but the imagery capacity, I'm being told to be brief, so I'll be quick. From a technical point of view, information that is placed in our systems nowadays, I brought a classical classification of software system and the current level. We have binders for documentation. We also have documenters, instruments where the nurses can add information, and there's also something on the reorganization of data, the graphical representation of data. But for level 4, 5, and 6, we are lacking. What the market produces today is almost useless. I accepted the challenge from Diego Bava to put together the private clinics or the clinics on certain topics, but it would be more than enough to start using the same products to insert in IT systems. I will just go forward. We're going to fill in the data and try to center them on certain diseases and see results on the same disease, but the instrument has to be easy to use and set up. Otherwise, whenever we make an inquiry, it's very difficult to work with it in order to improve it, because it asks for extraordinary commitment. Each of the facilities, big and small, we have the Sandoma group, and hence we have great big ones, but they are in trouble when it comes to every single sector, to explore them, to insert protocols, update them, keep them updated, and share them. If within that we had sort of a unique stakeholder that can organize software in an easy fashion that would communicate amongst them, we could use the possibilities that IT sector offers today. The clinical work that sometimes is subject to mistakes for information that is lost or that is not underlined in a sea of information that one finds. I just wanted to suggest this tiny item. Of course, we can add the technology that can bring more data, the home assistance part, the devices there offer loads of data, but we don't know if they have any clinical value. Nobody has ever organized the data, whether, for example, following the cardiac rhythm of patients. Is it useful or not? We can use algorithms to simplify data. There's a lot of work behind it, and which is bound to explode, but the operators themselves are not involved. The health part, the engineer part, I apologize. Thank you, Salvatore. Grazie per
into the challenges in your everyday's work. So I would like to go further with the session. So we have a next speaker is Ennio Tasciotti. He's a founder and director of the Center for Biomimetic Medicine at the Houston Methodist Research Institute. He is a director of the Center of the Musculoskeletal Regeneration at the Houston Hospital. Also, we know he's our ex-colleague, dear colleague. His discoveries during his rich research years resulted in 12 international patents of nanomaterials, biomaterials for biomedical use. He attracted many millions of funding from Department of Defense, NIH, and so on, and he's a founder of 3 startups. So I think that he has a really good position to tell us about his work, about the challenges during the COVID time, and what will be after COVID. Ennio, floor is yours.
Thank you, Natasha. Should I speak in Italian or in English? What's the rule?
As you wish.
Ok, well, facciamo in italiano, visto.
Let's go for English. The slides are in English. First of all, I'm terribly sorry that I wasn't able to be there in person, but I was tricked by airports. Traveling internationally is quite a mess nowadays. I want to tell you about what we did in the lab in the past 6 years on nanoparticles which are biomimetic. And this because my lab has always been interested in studying the biological barriers that are hurdles between the drug or the nanoparticles that are contained in the drug and the target site. These are biological barriers of various nature and complexity, vascularity itself, and all the immune system that is within the tissue, but also the endothelial barriers and the membrane, cell and nuclear membrane. Imagine a drug which is injected or it's taken orally. In order to arrive to this target site, has to go through all these barriers, which is quite an obstacle course. This renders the design and the engineering of a nano particle that can have a true target. What we did is study a universal system of nanoparticles that could target all the sites in the body which are affected by an inflammation, and this by stealing from the tricks that from the biology of the body, especially the immune system body, white cells, for example. While we ask a nanoparticle to arrive in a target site, we ask something impossible because it's 100,000 of blood vessels in the body. So there is an infinity of possible parts for a nanoparticle. And yet there is something that allows us to identify the areas, the tissues where we want our drug to arrive. This because the inflammation is part of the universal language of the body, which says something is wrong. It's common to a hallmark of pathologies that are of interest to us: tumor, cardiovascular disease, neurological disease, immunological disease, and metabolism disease as diabetes. The idea to use inflammation as a target and to create a nanoparticle that could identify it was a sort of key to open various gates. And we did this by studying the immune cells, which are already good at doing this. They find a way to find the inflamed endothelium, and through this, the process that is described in molecular biology in the past 20 years, they interact with the endothelium, and through the receivers on their membrane, they allow for the opening of this membrane and penetration. We concentrating on the property of the cell membrane, on the wealth of information that this membrane have at protein level. We studied all the proteins that exist on the membrane of these cells. We define them in various ways. We're going to be very quick here to avoid technical terms. This complexity that is on the cell membrane is also there on the nanoparticles which compose the cells. I work on biomimicry, and I always look at what I can steal and copy from nature. So our cells have been working with nanoparticles for before us. They are around 100 millimeters, and they are way better because they have a wealth of information both inside and outside, and we try to mimic them synthetically. This particle was debuted in 2016, and other work has followed suit because it has become a versatile platform. We isolate the cells from the patient, we purify the membrane from the membrane, we extract the needed proteins, which are re-including through a microfluid process, in a very easy way, repetitive way. We inserted in this nanoparticles. We can have chimera nanoparticles using very difficult proteins. We presented the leucosomes, which are half leukocyte, and then we had neurosome, cancersomes, endosomes, all a series of particles which have been patented and have already been purchased by some companies. Why? They can be standardized and created very easily using microfluids and using more different types of proteins. We can also upload all the medicines or drugs you can think of, because the liposome allows to charge molecules of any type. You can have a liquid core, or you put lipids and nucleic acids. We managed to create this range, a wide range of particles using both different proteins and different charges. What did you do with it? Since was a COVID session, I extracted, I apologize for the noise. I will mention some COVID work we carried out. This is an article from 1 year ago on SES Nano, where we described all the possible applications of nanotechnologies on COVID. And we got the article of the year. It was quoted many times. We can do a lot with nanoparticles and nanomaterials for COVID. First of all, because we can block a series of molecular events that are part of the virus cycle of life, and this within the body. But also because you can create materials that allow the blocking of the entering of the virus, creating more technological masks, for instance. Or even, we wrote this last year when we didn't have a vaccine, but you saw the use of nanoparticles to create vaccines, or to create nanotherapies that can boost the immune response. I'll talk about this in a while. This is what my lab is working on. This is another work we published where we used these nano vesicles. We managed to interact with the immune system and with the endothelium in order to increase the secretion of cytosines, the cytokinin, storm.
Di per sé, senza neanche un carico terapeutico, sono in grado di modulare la risposta del sistema immunitario. In questo era una COVID-like syndrome che abbiamo ricreato con LPS, quindi una specie di modello di sepsi, in cui gli animali muoiono in frettissima, cioè in un giorno, in 24 ore, gli animali non trattati muoiono. E come potete vedere, invece, gli animali trattati con la nostra preparazione dei leucosom caricati con dexametasone ne moriva solamente un 10%, n è uguale a 10, quindi moriva solo un animale su 10 del gruppo, mantenendo invece in vita tutti gli altri. Quindi una risposta molto importante, e questo perché studiando poi con l'Intravital Microscopy, che è una delle tecnologie che abbiamo sviluppato nel mio centro, quindi andando a guardare proprio come queste particelle si muovono all'interno del corpo, vi faccio vedere solamente questo per motivi di tempo, riusciamo a far accumulare, qui questo qui è, vedrete qui, questo è un alveolo polmonare, vedete in blu è il sangue, questo è live, quando iniettiamo i leucosomi vedrete la quantità di leucosomi che riescono a legarsi alle pareti infiammate dell'alveolo e quindi teoricamente a rilasciare sia il loro potere terapeutico attraverso il farmaco sia attraverso la loro attività di modulazione. Salto alle conclusioni, ma di base la mia idea è di pensare alla biomimetica come un sistema per creare nuovi sistemi di rilascio del farmaco e di interazione con il sistema immunitario. Per farlo devo ringraziare il lavoro di tantissimi ragazzi che si sono avvicinati nel mio laboratorio. Questi sono quasi 10 anni di lavoro in realtà su questa piattaforma tecnologica, e a parte il mio team anche tutta una serie di collaboratori in giro per il mondo che ci hanno aiutato. Grazie, scusatemi per il casino, sono seduto fuori l'aeroporto di Fiumicino e c'è un casino della Madonna.
Grazie mille, Ennio. Very interesting presentation, and thank you very much for all the effort to be with us this evening. So I would go further, and our next speaker is Kevin Eigner. He's a biotechnology department manager at Serichem Torviscoso in Udine, and this department is embarked on the program of biologics, biosimilars, incorporating microbiology, molecular biology, fermentation, bioprocess engineering, also for the coupling of chemistry to biocatalytic strategies. So, Kevin, the floor is yours.
Okay, thank you for the chance to speak. I'm going to talk about only 2 years of work, so I'll be able to speak more slowly. So I'd like to talk about the adventure at Serichem. Serichem's a fine chemical company which has introduced biotechnology in the past 2 years. The reason was to create this synergy between the chemists and the biotechnologists, especially in the major project, which is normal and long-lasting insulins. We've been working on the process, ancillary products, which also are very useful. We've been fighting against the intellectual property rights that are present in the field, but which actually are very short-lived. The biggest problem we have and we are working on is the property rights in the field, which never end. And we have to do this all with an eye on cost savings. So I'd like to sort of update you on what's been going on in the past 2 years and this synergy with the chemists for insulins, and also we've been working with some biocatalysis for other drugs. In particular, one was mentioned before by as a repurposing, that is the miglustat. So the verdict from our chemists about the synergy was biotechnology is easy. This is good news for Bruno and Natasa. You don't have to worry, you don't have to work so hard, because this is what's become clear from our chemists. And I think it's interesting for the general audience. Why is this statement? Why has it been made? And I think one of the important things about biotechnology is the huge number of tools and kits we have available now. In my experience, which has lasted much too long, a lot of these tools were not available, and now we really have access to a tremendous assortment of helpful aids. In terms of the high-tech, this is a high-tech meeting. My perspective on high-tech is to use high-tech to create applications which are low-tech, since cost is always going to be one of our major goals. Lowering the level of technology, making it more easily applied, reproducible, and accessible is a major goal. One of the other things we have to do, we're able to do, are screening approaches. In biotechnology, often we don't have what we want, but we have a large pool of options, and by correct questioning of the system, we can actually find out what we need. We can create our own tool. Some examples of this are, for example, microbial prospecting. For example, if you want to do a conversion of a biomass, if you find an animal, insect, something that eats that biomass, you have a source of the enzymes that can process it. In addition, we're also doing this in Styrichem, microbial evolution. If you have a catalytic system which works at some degree, you can create artificially an evolution and modify and create your own catalyst. This is also not to exclude the bioinformatics and artificial intelligence approaches. All right. Let's see, this would like to go forward, I hope. It's not going forward. Here, okay. So there's always a negative side. The older people in the audience will recognize flip side. It'll probably be the last time it'll be used in the English language. What are the problems that we run into with biotechnology and the application of these tools? In particular, the time it takes to do this work, the adjudication of success. When do you know that you have gone far enough? For example, when we make clones for insulin, in the past 2 years, we've made probably 700 different clones from Pichia pastoris yeast that produce insulin. How do we know that we've gone far enough? There's a neighboring company not too far away, which can easily do 7,000 clones. And we've talked to other company in India which has done 70,000 clones. How do you know when to stop in these approaches? We're working with living organisms, so living organisms have stability issues. Are they going to remain as you think they are? Are they going to be productive over long periods of time? The difficulty to guarantee success and also the dependence on these life forms is quite a complicated event. I was asked recently, what are the variables involved in the growth of a microorganism? And I would say probably infinite. So very hard to predict, very hard to model. It's not going to be a simple mathematical equation. All right. In terms of biotechnology in times of COVID, the supply chain has been difficult for us. Instead of 18 days for a piece of equipment, it usually takes about, now we're getting 18 weeks, would be something which would be quite quick. We're dependent on high technology instrumentation, and especially, which is very critical for us, is plastics for sterility. These are very difficult to get, and they're causing some problems for us. In terms of APIs, active pharmaceutical ingredients, this is what we want to produce chemically and also biotechnology. This is the main goal, is to produce API processes in production in now. Post-COVID, well, pre-COVID, for these drug supplies, there were three main factors: cost, and cost. All right, and this led to the outshoring of productions of API to India, China, and other places, and we had a big loss of the production of APIs in Europe. Now, because of the supply, we have major problems in availability and also the sustainability, which we would have to do here in Europe, and is not necessarily applied in other places. An Indian chemist asked me, do you really have to do something with your waste? Yes. We have to do it, and apparently they do not. And this, again, comes back to cost. In order to have a more available and sustainable drug supply, inshoring is extremely important. All right. I'd like to just take a moment to talk about the healthcare utility model. Also because as an American, I can finally say something positive about the American health system. This is the example of Civica RX. It's a nonprofit company that's addressing inshoring problems. And what they're doing is they're buffering the distribution events, the increases in shortages of the materials. They're doing, they're performing quality analysis, and also applying some, should be applying some sustainability requirements. This project started in 2018 because of the inadequate supplies of antibiotics, so it's pre-pandemic. And so this was, this is an approach to develop a system for inshoring, which is being successfully applied. One thing that needs to be recognized is this whole system of inshoring increases costs about 30%. All right. So if we try to have a system which is designed to improve inshoring, and yet we're going to go back to the old system of judging the API and the consumption by pharmaceutical companies or clients, patients, and if that the judgment of purchasing is based only on cost, this is not going to be a successful model. So being able to incorporate these other important aspects is fundamental. An example are the generic drugs. The generic drugs have APIs, and even many brand name have APIs, and which are unreliable. So even this is a major issue that needs to be addressed in the future. Thank you.
Thank you very much, Kevin. And I would like to proceed and ask Giuseppe Patuelli, Marketing and Sales Director of the Pharma Division BioValley Group, and he will outline their work and experience in rare disease drug market and their results obtained in the first 2 years of presence on the national market. Giuseppe, floor is yours.
All of you. I will proceed in Italian just to be in alternance with Kevin. Dunque, allora, la mia presentazione
My presentation changes what you've seen so far, because I will refer to something very different from research, but still related to it. And I'll try and explain. I hope you can see my slides. Not yet. Waiting for the slides. You should see them now. There you are. Now, very briefly, I will talk about the impact of COVID on our situation as a BIP Pharma. Now, BIP Pharma is a young company. It's a pharmaceutical company that was set up in April 2019, focusing on the production of orphan drugs for rare and neglected disease in Italy. We are a small group of professionals with a long-standing experience in other international pharma companies. And basically, we have experience, especially in managing complex hospital problems, not just in terms of marketing and distribution, but managing the complexity having to do with the clinical studies and the clinical projects, as well as the introduction of new innovative drugs and their admission, authorization onto the market. We basically have two products. The first is miglustat, is the only approved therapy for a very rare neurological disease, which is a Niemann-Pick type C disease. And we also have Ciclus. Ciclus is a very particular hydroxycarbamide formulation, which is the gold standard therapy to prevent vascular occlusive crisis in sickle cell anemia. Very briefly, I'd like to mention these two products. Regardless of our normal activities as a pharma company, we are also active in the field of development projects. And for miglustat, we are developing, together with two centers in Rome and Pisa, two projects aimed at enlarging the indication of this important molecule to other rare neurological diseases, lipofuscinosis and spastic paraplegia. Ciclus is again an innovative drug because it allows, unlike what happened in the past, it allows to accurately establish pediatric doses. It is a very important medication. It is a very important drug because it improves dramatically the quality of the lives of these children and their families. And the drug can be adequately taken, which translates into a greater effectiveness and safety. What was the impact of COVID-19 and what did we do? For those who are used to working with hospitals and health agencies, and hospitals were very busy, we know, during the pandemic to manage the pandemic from all viewpoints. Well, our commitment was to ensure the continuity of treatment for our patients. Many of our counterparts, many of them physicians, called us. They got in touch with us to ask about the possibility to ensure continuity of treatment. Rare diseases usually affect patients that are already at risk. I'm just back from an Italian conference of the Italian Society for Hemostasis and Thrombophilia, and there we talked about sickle cell anemia, and one of the data relating to the pandemic is the increase in the number of deaths among pediatric patients affected by thalassemia and sickle cell disease. So this was a very serious problem for our counterparts. Nevertheless, we've been trying to support not just physicians, but also patients' associations and patients' families, supplying documentations on the relationship between COVID-19 and the diseases that we were treating in an attempt to support them. It was mainly a psychological type of support, but we wanted to ensure the continuity of treatment and the safety of our treatment. As for the supply of our drugs, as Mr. Enger was saying, we mustn't forget that COVID made it difficult to find APIs and as well as many raw materials for the manufacturing of drugs. And we were affected as well. So together with our suppliers, we've been trying to implement different actions to ensure the continuity of our supply to hospitals or to the families, thereby avoiding the discontinuation of the treatment, which could be very dangerous. And during the COVID-19 pandemic, we also had the opportunity to supply Miglostat for research purposes, to verify whether it was an effective antiviral drug in patients affected by COVID-19. Today, we are restarting. The pandemic has taught us many lessons on the management, on our relationship with hospitals and physicians. But what was important for us is that we succeeded, thanks to contacts that we were able to keep during the pandemic, is the relationship that we have established with new international pharma companies specialized in the research and production of of orphan drugs for rare diseases. And next year we will launch two new products for metabolic diseases. This is an important point of arrival for us, because very likely we will be able to operate on the international market, in particularly in Europe, and therefore we will be able to interface with our larger competitors. Our team has a focus on the market, on sales, but of course we are always ready to respond promptly and timely to new challenges. And the challenge posed by COVID-19 was indeed quite an important one. Nevertheless, we succeeded in improving our own situation and in responding to the demands that we received. And I hope that this lesson can teach us to tackle other challenges in the future. So that was my contribution. Even for a company working on the market, COVID can be a challenge, but an opportunity at the same time. Thank you.
Nice last sentence, that yes, it can be a challenging time, but also the time of opportunities. So I would like to call next speaker. It's Sara Raccovelli. She's an industrial researcher in the Transactiva, and in her intervention in Udine, a company in Udine, in her intervention she will briefly introduce plant molecular farming as an alternative and interesting strategy to produce biopharmaceuticals by employing whole plants and vegetal cell culture bioreactors, as already Dr. Bembi mentioned before. Floor is yours.
Thank you. Thank you for having me. Parlerò in italiano se non è un problema.
I'll be speaking in Italian if that's not an issue. I will talk about green biotechnologies and how these can be an important resource in crisis scenarios as the one we are facing now. In order to do that, I need to introduce the plant molecular farming, which you have already done partly, and it's the use of full plants or vegetal cells in order to produce therapeutic proteins. How does this process happen? It starts from a protein with a therapeutic aim, you transform it in vegetal tissue, which then produces this heterological protein, which can produce a drug. This is a technology that requires a lot of cross-cutting competencies from medical biotechnology, vegetal and farming techniques. But why use plants when we have fermentators for mammal cells, which work very well, but there are, of course, some intrinsic advantages of the vegetal cell. I will stop on biosafety because there are no cross transport between the vegetable system and viruses. You can't lose product batches through contamination with human pathogens, and sustainability, both from an economic point of view, because there's reduced costs in the upstream, but also environmental, because they naturally fix carbon. And this can be a part of a circular economy that can be extraordinarily important in this period. What is the groundwork in plant molecular farming? Is to substitute the classical mammal cells, which are quite expensive to maintain and to produce, with something a lot more simple, with greenhouse, for example, where the natural resources are being used. The bioreactors are the rice seeds here. Is it possible to substitute really the bioreactors from mammal cells with vegetal cells, plant cells? Yes, in following the plant system, starting from the host species. You can virtually choose any plant which can be transformed and produce heterologous protein. But algae, moss, and others, you have various transformation methods. I would like to quote a stable one which produces a GMO, which is a plant that has information stable for this heterologous but also transitory ones that do not create GMOs but can produce a little bit of protein, which happens with the mRNA-based vaccines. We don't become a GMO. The proteins that can be produced are virtually wide protein complex, which are not part of the vegetal world, but also complex proteins and semi-viral proteins. I want to talk to you about one of these. Particular combination can be very interesting. I want to talk to you about the transient transformation of the Nicotiana benthamiana plant, which is a transitory transformation, no GMO involved. The plant is transformed temporarily to produce semi-viral particles, vaccines, and monoclonal antibodies. Why this technology? There are various advantages. The main is speed of production. With a facility and equipment that is working already, you can have few weeks from the beginning of the project till the production of proteins which are ready for clinical trials, not months. Scalability, which is a part of the plant system. A plant is a bioreactor. In order to create more facilities, you don't need very expensive plants, but just in a green chamber. This method also offers great quality and accuracy in the sequence that is introduced, and the error that can occur in this protein is close to nil. This system is furthermore very versatile, because if the equipment already works, can switch to another molecule without using further investments and in a few weeks. Hence, this technology exists for many years. It has always discussed in a theoretical way in academia, and I want to show you some examples, concrete examples, of how this technology is being developed as we speak. For what, namely? For a crisis situation like the one we are dealing with right now, thanks to the characteristics I've mentioned. We noticed how the pharmaceutics during the outbreak of Ebola in Africa created monoclonal antibodies against this outbreak. It was approved temporarily, and it was approved by Medicago, a Canadian company which is producing vaccine based on viral particles, and not only in the third stage, but also waiting for the approval for a vaccine for the seasonal flu. Another company, AV, which has another potential candidate for COVID-19. I would like to talk about Medicago because it's the company which is closest to the go-to-market stage. What you see is a facility of production where plants are transformed in a transitory manner in order to obtain a green mass to extract the vaccine, and the beginning of the process, pharma-grade condition. We have 5-6 weeks in the pharma-grade conditions. It's real, it's happening. And from this, I would like to speak about an Italian case where we have a synergy of local excellence that are working together to apply the same system here in Italy. Transattiva, which works on plant molecular farming, and the Department of Biomedicine and Prevention in Tor Vergata, Rome, started this project involving the institutional interest in Trieste, such as the university, to build a green platform based on this plant molecular farming, which has to produce antiviral vaccines. Our initial target is, of course, COVID, to follow on the wave of information and funds that are available. But our plan is that to create a platform that will then, thanks to its versatility, can be used for future crises, hoping that we are not going to face many. Very quickly, I will talk to you about the stages and current progress. We identified immunoreactive peptides that have been, we designed some VLP-based vaccines that bring the information to service, and thanks to computational biology, we predicted the stability of the particles in silico, and we saw, which was of course the best were designed and manipulated in vectors. We had a transit transformation of the tobacco plant, and then we produced the first viral particles, VLPs, without COVID antigens, but in these weeks with COVID antigens, and we are producing them. We're doing this. Final perspective on the future with data from the medical studies and all the groups that worked on this type of technology. The protein yield and VLP amount and biomass yield, it is possible with a single facility with 2,500 square meters surface can deliver each 3 months doses to cover the entire Italian population. And I hope this will become a local reality. Thank you for your opportunity. Thank you, Sara. We also hope so. The next speaker is Elisa Margotti.
Department in Halifax, in the company here in Trieste, and she will talk about the acquisition by Halifax of the fully equipped protein science facility present in Area Science Park here. So please, Elisa.
So, as there is at least one speaking, I think I will hold my speech in English because I prepared it in English. So, good afternoon, everybody. My name is Elisa Margotti, or Eliza, as I'm usually known in the worldwide lab. And today I would like to tell you a story about how to raise an opportunity out of a crisis period like that one that we have been living since the beginning of last year. And at the beginning of this story, we have Halifax SRL, a company settled in Padova, near Padova, with more than 30 years of experience in the market of diagnostical tools and medical tools. And Halifax has 2 research and production sites here in our region, in Friuli Venezia Giulia. One in Nimis, close to Udine, and the other one here in Trieste, in the campus of Basovizza Varia Science Park. This one, Halifax Research and Development here in Trieste, is the youngest one. It was opened in 2017, specifically to develop a breakthrough in a qPCR system. And in fact, we are talking about this molecular mouse system developed in a partnership with ST Microelectronics. That is the world's first handheld qPCR system. As you can see, it's compact, easy to use, and fast. And at the beginning of last year, the whole team of Halifax Research and Development here in Trieste really promptly reacted to the emergency determined by the COVID-19 pandemic. And before the end of the year, we were able to develop and launch the molecular mouse platform, meaning the instruments, and the cartridges dedicated, and the software that controls the instrument and analyzes the results. Moreover, we were able to set up a production facility for the production of cartridges in order to meet customer need. I am really sorry, but the formatting of the slide is a little bit lost with respect to what was meant to be. So together with the increasing request of tools for the molecular diagnostic tests, the need of rapid tests able to detect the presence of SARS-CoV-2 antigens and afterwards also neutralizing antibody have been taken by Halifax as a further opportunity to add new expertise to the know-how that was already present in the company. And with the intention of becoming as much independent and autonomous as possible in the development of rapid test, last year, Halifax R&D has acquired a fully equipped facility that was already present in Area Science Park with the whole set of instruments and technologies to follow the whole process of selection and screening of monoclonal antibodies and production and characterization of recombinant proteins. And together with the facility, Halifax also hired a team of extremely skilled people, and luckily I'm one of them. So a team of skilled people able to design, develop, produce and deeply characterize protein of high quality and also low cost, or at least relatively low cost, especially considering what is already present on the market. And of course, at first, this protein science team was meant to integrate in Halifax core business in the market of diagnostic tools which has a focus in microbiology. But if we enlarge a little bit the point of view, of course, the target of this protein science team could be virtually, and I stress the word virtually, any protein, and to span also a wide range of fields of application, for example, development of rapid test, both diagnostic and prognostic, and also ELISA kit and therapeutics, so therapeutical biomolecules. So in order to describe a little bit more in details the kind of expertise that Halifax has brought in-house with the acquisition of this facility, let's have a look to the workflow that leads to a very well characterized final protein. So at the beginning we have the screening and selection step. That means that the construction of antibody libraries in single chain or Fab format that we use to select for the selection of fragment monoclonal antibodies with the technique of phage display. And then, in case of need, these fragment monoclonal antibodies are then reformatted in IgG. In case of other kind of recombinant proteins or antigens, for example, we design the most appropriate expression construct for these antigens and the recombinant proteins. Once we have the construct, of course, we go to the expression of the proteins of interest, expression that can be done both in eukaryotic and prokaryotic cells, because we have two dedicated and separated facilities for these two kind of cell systems. And of course the process can be also scale up, because we also have a bioreactor in our hand and we can scale up this process. Afterwards, of course, the protein needs to be purified, so we have the purification step. We set up the best purification protocol for each specific antibody and antigen we are dealing with, basically through FPLC-based chromatographic step. So we have all the kind, of course, of chromatographic step: affinity, ion exchange, hydrophobic interaction, and size exclusion chromatography. And with our purified protein, we go through the characterization, deep characterization. So we investigate the biochemical and biophysical properties of the protein. So protein integrity, purity, the oligomeric state. This in particular is performed through size exclusion HPLC. And then, of course, the binding affinity, very important when we have monoclonal antibodies, the affinity for its antigen, but also in case of other kind of protein, for example, the interaction of one ligand with its receptor. And the binding affinity is investigated both by ELISA, giving them an information, for example, of EC50 concentration, or with Biacor X100, an instrument that allows to investigate more in details the association and dissociation phases of the binding of these two proteins. And then we can study as well conformational stability with the differential scanning fluorimetry, and more in general, of course, we can follow the stability of our proteins along a period of time, months or years, according to the need, and also considering different storage temperature beside the different time. And all of this, of course, brings us to our goal, to the finish, to our characterized protein. In the end, a glance to the future. Of course, the protein science team will go on producing antigens and antibodies to improve the development of diagnostic and prognostic tests implemented and commercialized by Halifax, trying to meet the needs of the market, especially, as I was mentioning, the microbiology area. But of course, we are open to study other pathologies, for example, in the oncology or veterinary field. And also, as I was mentioning previously, we could also develop pharmaceutical, therapeutical biomolecules to be delivered as API, as active pharmaceutical ingredients, to pharmaceutical companies, of course. This has to be seen as an activity in support or in collaboration with experienced pharmaceutical companies, because, of course, Halifax has neither the structure nor the experience to develop further than the stage of API any therapeutical biomolecule. But in this sense, we are willing to support other companies and to get also support from this company for the first development of this kind of molecule, and also to have some suggestion about potency assays that can be set up. And in the very end, here you have the whole team of Halifax R&D here in Trieste. And I really would like to mention all of this person, because it's true that today I'm here speaking in front of you, but I'm aware that all of you know that behind the 10-minute speech there are months or even years of hard work and dedication. And I really thank all of these people, all of my colleagues, for everyday support and collaboration. And thank you very much for your attention.
Thank you very much, Elisa, and it's really good to hear that Halifax recognized the need of the local production, and I think that COVID helped to realize.
This was really some kind of rescuing operation that was really important, because otherwise all the expertise and the know-how that was really present here in the territory would have been lost, because this facility was missing, basically. So this would have kind of a big loss for the area, in my opinion. So thank you.
I think we are also here to present what we have, which skills and the richness we have here locally that we should not lose. For sure. Thank you very much. So I would give a floor to the Faustino Ferrazzi. He's chairman of the Serichim Terviscosa, and his short presentation will focus on the role of the company Serichim in a post-COVID scenario where there will be many funds, European and regional, available for research and innovation that must be exploited to the best to achieve sustainability, eco-compatibility objectives set by European Union.
Thank you, Natasha. Although my presentation is in English, if you don't mind, I will go on in Italian. Devo dire che mi sento un po' a disagio, perché io non sono uno scienziato.
I feel a little bit uncomfortable because I'm not a scientist, and I've seen some really interesting and beautiful things that were presented by scientists. I would like to underline that I tried to underline it here in my presentation. I wrote EuroBioHighTech NEC 4.0 next to the other logos, because I believe that one of the important things that have been underlined from today's presentations is that there are so many opportunities, ideas, opportunities here in the region. Many centers of excellence, not big companies. So what I want to say, and it is important, and the message that I would like to convey today is that we have to work as a network. I represent a small company that carries out clinical research in chemistry, the chemistry of processes. We are available for other points in the network to develop the innovations and the ideas that are created from centers of excellence. Another element that I wanted to underline, I'm going to be quite short. Sedicim was presented various times. It's headquartered in Tor Viscosa. Kevin said that before, the chemical research of processes. And when I mentioned this sort of type of chemistry, something that is pervasive in all industrial sectors. Whoever has a process or has to deal with chemistry and biochemistry. So I'm not going to be too long in telling you what we do. We can be a tool, a sort of a screwdriver to use by anybody to accomplish their innovation project. I would like to remind you the context that we have been operating in, the COVID situation. What emerged due to COVID, especially in the so-called developed countries? The fact that we are not independent. 90% of raw materials for drugs, for the most common ones, paracetamol, antibiotics, are produced in Asia, China. We risk lacking the basic drugs for our health when a geopolitical decision interrupts international trade. So what we must do is try to bring back to our countries the essential type of production. And we can do this through innovation. We can't bring back to Europe or to the United States what we already outsourced in 2001 to China and India because it was too costly to do them here. The environmental impact was too high. So innovation is key to reach this objective. And luckily, decision makers have realized the same thing. In all the position papers and all the political declarations that have been done at European or international level, we see, we see that the decision maker finally understood that they must support these innovations through an injection of funds. That's why I brought a couple of figures with me. One regarding research in the following 7 years, the the EU with Horizon Europe allocated 95.5 billion euros for innovation. And if we are to look at these 95 billions, we have national funds which will be added and used. If we look at Horizon Europe and its 3 pillars and 6 clusters, 4 directions, there's a full explanation by APRE on how it works, but out of the 95 billion, 8.25 billion are dedicated to the health cluster. And this is something we need to bear in mind, because it's 8.26% of all the investment carried out in Europe. I'll finish preaching by saying that, especially in a region such as the Friuli-Venezia Giulia, where we have a lot of SMEs in innovation, we can only succeed by working together, trying to connect the various competences and availabilities here in this area. I have in front of me 2 examples. We have worked with the institution on the insulin technology, and Natasa and her team have created and designed. We want to offer results in research and transform this research in a product which has good technology behind it and it's competitive. We want to offer this technology to developing countries, but not only, because from the point of view of health, we risk ending up to be developing countries too. Another example is Bruno with his Transativa. They will bring forward a project that I believe in, even if I'm not a scientist, mimicking with plants, producing with plants the active substances that your healthy body can produce on its own. If not, you need something outside the body to produce them. To end up with a drug, you need chemistry, you need sanification, purification, lyophilization, and we are there to give a helping hand. We believe in this project. We invested, thanks with the team leader, and the investment in partners must not be forgotten. We invested heavily, a lot of resources to reach this objective, and we are also committed in this second moment for big European projects that want to put into practice this declaration of intent, producing drugs in a green way, sustainable and competitive drugs. So I would like to wish you and all of us to be courageous, determined, and willing to reach this objective, that is independence of our country and our region. When it comes to possible other pandemic situations, or just the flu that might have an impact in the future. Thank you. If I was too long, my apologies. Thank you for your attention. We created a new slogan, Serikim. It's a tool that creates sustainable solutions that bring innovative ideas to the market. Thank you and have a good evening.
Really good slogan. So with this, we will close our session, and I was asked to give a quick summary of this session, so I would just go through all things that we heard here, and then maybe if we can have a presentation. I have 2 last slides to show the perspectives and what will be after COVID. So today, during this very interesting session of research and innovation in biotech, we heard from ICGB that responded very quickly to the emergency for the biotech point of view by sequencing and of the genome of the virus, and also developing different diagnostic tools and helping the companies and the institutions. We heard that Swiss Integrative Center for Human Health in Freiburg also contributed to the diagnostic by patenting method for the molecular diagnosis in saliva to be used for COVID-19 infections, and we hope that it could be applied in the future for the chronic disease, other chronic disease. So I hope that we will have we can apply all the lessons learned during the COVID. We heard from a representative of Sanatorio Triestino how they respond, of course, very responsible action during the pandemics, how they struggle to apply and choose different test technologies, diagnostic tools during the COVID-19, and how they collect data and they presenting the need for the integrated approach that could not just collect this data electronically, but also suggest possible therapies. From the representative from the Center for Biomimetic Medicine in Houston show us very interesting solutions for biomimetic nanovesicles for targeting inflamed vasculature, but also presented a very innovative approach of leucosomes and drug delivery and nanoparticles that could be applied and was applied during the COVID pandemic, but also could be applied on any other medical condition. We heard from Kevin and from Faustino that Serikim as a biotechnology department, they're very active in the production of biologics, and in the post-COVID scenario, they really are keen and suggesting that there are so many fundings from European Union that is really good to hear, and they also emphasize importance of the local production. Transactiva is working actively to build a green platform and plant molecular farming for a rapid and effective antiviral vaccine. So that was their response during the COVID, but also not just for antivirals, we heard for other biologics. Halifax, we are really happy that these skills are not lost. So protein science department as a new department with the Halifax will be able to enable recombinant protein facility production for antibody screening and rapid test development. And we heard from BIP Pharma Division, BioValley, that they're very active in the field of the rare disease and the orphan disease, even in these very challenging times. So if I can ask the IT unit just to put my presentation. I have just last slide that deals with, is it possible? I just need the... So just like to conclude, sorry, just to go at the end, sorry. Can I go back? Yes. So after the pandemic, we heard how we were really very active during the COVID, and what we need to do, I think that all of us will agree that we have to preserve what is currently in place. So we demonstrate in biotech companies all around the world, they demonstrate they're able to quickly increase manufacturing capacity, quickly respond to the needs, and that biotech is the field with the faster than ever response to emerging health threats. Then the pandemic has caused all of us to consider the importance of things like early disease detection, health and wellness, anti-infective drugs, drugs, remote care, and so on. And we mentioned more than once during all the presentation that effective responses demand international, not just national, regional, but also international cooperation. And as we heard now from Faustino and many other speakers before, we all agree that invest in a larger network of partners that can quickly ramp up local manufacturing capacity is the key. So we have to ensure national or regional self-sufficiency in the production of we heard the different essential goods, diagnostic kits or different tools. And it is important to have abundant local capacity in the terms of the laboratories, infrastructures, experts to respond effectively to the challenges posed by this pandemic, but also consider the risks maybe in the future outbreaks. So after the pandemic, when innovation and research comes too, everyone agrees that this shorter disruption in the healthcare innovation, maybe in the other fields that were not connected to the COVID, will be temporary. So the new innovative drugs will continue to be generated very fast, and really applying the lessons learned, how to use and how to fast produce more than ever this personalized medicine, different therapies, gene editing. So there is a really hope that this will be very fast growing field and growth of the personalized medicine. So I would just like to finish with these two sentences that is emphasize all the experts around the world, and we today agree that there is a need for countries to implement measures to protect their innovation system as part of their stimulus and recovery packages, and scale up funding for innovative startups, regional approach, additional and long-term financing for local scientific capacity. I would like just to ask Bruno if you have something to add.
I don't have, you have covered quite all. Just an observation to consider something very new that the pandemic has demonstrated, that the great companies has failed. In the run to the vaccine production, you have now two new companies, BioNTech and Moderna, that grow up and are on the market. Okay, that means that the necessity of scientific research company are essential. And if we consider in our region, we have 8 biomed academic departments that every year graduated about 400, 500 young people in a very good, very elevated, high quality capacity. So we must, our main commitment is to develop a milieu in our region to give to this young researcher a future, and more or less we have presented the possibility in our meeting today. Another important point, that means also the involvement of the so-called political decision makers, is to understand that all this need an important economical support. There's another point, just to permit to the network that is here present to grow and to improve. And my last observation is something very positive. European Union recognize the retard of the system to approach the very fast biological modification as pandemic has presented, and now they have accelerated all the process for recognition for arriving on the market of new products. So one of the commitments of the European Union is to renovate, to rethink, to think all the system that is very bureaucratic, that is today present, with which we have to afford when we want to develop a new drug, a new facility, a new diagnostic facility. So within the end of the year, probably they will renovate and rewrite all these procedures and accelerate it. And let me say, I don't know, I can't find how to say, to run and velocizzare, no?
To speed up.
Yes, they have to speed up all this process. And this is essential, is very important, essential to afford the new challenge that we will have in the future. So create a network, believe in our young researcher, and think a lot about the science and how to transport the science discovery to the market. This is something very important. More or less.
Thank you very much. I would like to thank to all the speakers and all of you for your attention. I think that we can close the session. Thank you.